Hi everyone, Fritz here. In today's video, we'll be going over your gram stain. The gram stain is a staining technique that we use to differentiate between gram-positive and gram-negative bacteria. The component of interest and the one that we'll actually be staining is called the peptidoglycan. And the peptidoglycan is the surface layer of the bacteria. And as you can see here with the gram-positive bacteria, it's actually thicker and exposed to the environment. The peptidoglycan on a gram-negative bacteria is much thinner and protected by the outer cell membrane, but also sandwiched in between another layer of plasma membrane. Here, we only have the plasma membrane behind the peptidoglycan layer on the gram-positive bacteria. Now, a couple of tips before we get into the steps of the gram stain is that you want to flood the slide. Completely cover it in the respective dyes for the respective amount of time. That way you'll be ensured that the bacteria takes in that dye. And then lastly, you want to make sure that when you get to the decolorizing step that you are very strict on your time here. Do not over decolorize your bacteria because otherwise it could skew your results. With that out of the way, let's jump right into the first step of your gram stain. The first step is going to be heat fixation. And if you forgot what heat fixation is, you can go ahead and check out the link right over here. Once you've heat fixed the bacteria onto your slide, you're going to want to add crystal violet. Crystal violet is going to act as our primary stain. As soon as you have flooded the slide and allowed the appropriate time to elapse, go ahead and rinse it, and then you're going to add iodine. Iodine is going to act as the mordant. And a question that you can expect on test day is, what is the role of a mordant? And if you guys need a tip, look up the crystal violet iodine complex. Once you've flooded the slide with iodine and the appropriate amount of time has elapsed, go ahead and wash it out with water. And now you're gonna add your decolorizer. 4 is going to be your grams alcohol. And this is gonna be that decolorizing step that you wanna be very careful and only do for 10 seconds. As soon as 10 seconds is up, wash it out and then add your safranin. As soon as you've flooded the slide with safranin, go ahead and wash it out and blot dry. Now you can observe your slides underneath the microscope. Once you have the slides properly orientated and under oil immersion, you're gonna see one of two things. You're either going to see the color blue or the color pink. Blue is gonna be representative of your gram-positive bacteria. The reason why is because the peptidoglycan, being so thick and has the exposure to the elements, is going to easily take up the crystal violet. And this makes sense because it is exposed to the elements. It should take up the primary stain. And even when we add the grams alcohol, that would shrink this structure because alcohol acts as a dehydrator. It would shrink it and squeeze some of the dye out. Even if we add the safranin and it takes some of it up, crystal violet is a darker color than safranin. So even if some safranin was taken up here, it would be covered up by the primary take up of crystal violet. However, if you have a gram-negative bacteria, it might take in some of the crystal violet, but because the peptidoglycan is much smaller and protected by this first layer of plasma membrane here, when you add the alcohol and it shrinks it, the already reduced size of the peptidoglycan is gonna squeeze out virtually all of the crystal violet. And because it was protected by some of it, it didn't get that much entry anyways. So it's going to shrink and squeeze out the little bit of crystal violet that we already have. That way, when you add the safranin, it will be able to color the peptidoglycan. So this is the purpose of the gram stain, is to determine which bacteria do we have. Do we have gram-positive, which will be colored in blue from the crystal violet, or do we have a gram-negative bacteria, which is going to have a pinkish color from the safranin? Another key indicator is that normally gram-negative bacteria are smaller than gram-positive bacteria. And typically, the bacteria that we're concerned about when we're studying diseases tend to be gram-negative. And make sure to write down the bacteria that you use for this lab because you probably will be asked on test day which of these bacteria, and they will give you the 2 different bacteria, is gram-positive and which one is gram-negative. And here are some additional questions that you guys can expect come test day. Now feel free to answer those questions down in the comment section below. If you found this video helpful, please leave a like and subscribe, and don't forget to check the description for additional study materials. If you have any other questions, feel free to leave those down in the comment section as well, and I'll see all of you in the next one.